that just give you a predisposition to being diabetic. If your exposure to sugar is relatively low your entire life, the chance of being diabetic is actually low, but if your exposure is very high, given those alleles, your chance of being diabetic is very high. Okay? So that's why it's hard to actually predict all the time for human diseases the precise chance that you'll get it. You can give probabilities. Okay? And it depends on the disease. Some things it's very clear, some things it's just an increased chance of suffering from a particular disease. Okay? And even to this day, relatively little is understood about the genetic contribution to most multifactorial diseases, though that's changing all the time. You probably hear in the news all the time that a gene for such and such disorder has been mapped, and it's usually a gene that contributes some fraction to that disorder. Most of the ones that are very clear that give rise to a disorder were mapped long ago, and most of the ones we see now are multifactorial diseases. Okay, so that does allow, though, for genetic testing and counseling. So you can actually do things where you have some idea of the underlying genetic basis for certain diseases, and so it is possible to give parents advice on what their chances are of having particular diseases. Okay, the hard part is, of course, knowing what to do with that advice, what you decide to do with that. And as we go along, we'll talk something about what you can do if there's any ways to intervene in those kinds of situations. Okay, and so the idea is that the advice is based on, again, knowing something about the way in which these traits are transmitted. Okay, so what's useful is to have a genealogy of the family, so you actually get some idea of what the genetics of the disease are and the genetics of the parents. Okay, and so that can help you predict the odds of that couple having children with a particular disorder. Okay, and probabilities are predicted on the most accurate information at the time. The thing is, we're always getting new information about how these genes are caused, what causes them, and how they're transmitted. So what can be told to people changes with time. Okay. There are a growing number of ways to test for carriers, okay? And we can talk about that a little bit when we talk about what the structure of genes actually is. But nowadays, you can hear that you can go in and you can be tested for certain things. Or how many of you have participated like in the 23andMe thing where you actually know some of your genotype? No, not very many. Okay. And so you actually can have yourself typed for a lot of loci, and you can get probabilities for suffering from certain things, what your chances are relative to the population. And they're, again, just probabilities. They don't define your future necessarily. Okay. Okay, so just to review then, okay, and we've gone through all of this, but just a table to quickly review the basics of the genetics we covered now. So we can get different relationships amongst alleles of a gene. So we talked about simple dominance, incomplete dominance, codominance, multiple alleles. We gave examples of all of these already. In pleiotropy, where a gene can have multiple phenotypes. So the sickle cell anemia is actually a case of that, where being homozygous has a downside and it causes the red blood cells to be sickle-shaped, it causes problems with circulation. But there's also another phenotype, which is that when you're heterozygous, you have an increased protection against malaria. So that's an example of a pleiotropic function of a gene. And then we ended the last lecture and started this one with talking about epistasis, where the sort of dihybrid cross we talked about is generating a 9:3 to 3:1 ratio of genotypes, right? Or phenotypes. But if we think about the ways that those genes can interact, we can get variations on that ratio. Okay? And so we had a question at the start of today's lecture. We showed a variation. We ended last week's lecture with other examples of that as well. Okay? So you should be familiar with that. And then you can, of course, have polygenic inheritance, which we just talked about, where you have many genes contributing to a phenotype, and because of that, you can get sort of a continuous variation in the trait. And we gave examples of skin color and height in humans as good examples. Okay? So we're going to leave the strict genetics now, and we're going to talk about how it is that when you generate gametes, you actually ensure that they have one copy of each chromosome, and what the consequences are of that and the way that those chromosomes sort for the variation you see in the next generation. So, but I want to emphasize that the best way to learn doing this kind of genetics is really to do problems. And so I'm actually going to post up 4 problems for you later this afternoon on the DSpace site. And again, they're not required, but I would urge you to work your way through them and make sure you understand them. Okay? And I'll post some more problems towards the end of this week when we've gone through linkage, and they'll include those as well. Okay? So we're going to move on now to talking about then how offspring really inherit chromosomes from their parents. This is chapter 13 then. Okay? So children don't inherit the physical traits, right? What they're inheriting is genes from their parents. So they're inheriting, chromosomes are inheriting DNA from their parents, and that's really the physical thing that they're inheriting, and then you get phenotypes from those, right? And so again, just to review, to make sure you keep in mind the definitions, right? Genes are the units of heredity, and they're made up of segments of DNA, and the next week or two we'll talk much more specifically about what makes up a gene, right? Genes are passed to the next generation via the reproductive cells, gametes, and so humans are the sperm and egg, okay? And each gene has a specific location called a locus on a particular chromosome, okay? So for your average gene, you have one copy on a chromosome, and because you have 2 copies of chromosomes in all of your cells, one from your mother, one from your father, you have 2 copies of that gene, right? And again, that's what we've gone through so many times now about the Mendelian pattern, that you have 2 copies, and you have 2 alleles, right? And then the DNA is packaged into chromosomes. And again, you only see chromosomes as condensed units when a cell is ready to replicate. Otherwise, it's usually uncondensed, and you don't really recognize it as a chromosome if you look into the nucleus. Okay? So you can have asexual reproduction, meaning that a single individual can pass genes to its offspring without fusion of gametes. And so these are clonal populations. We talked about the simple case of bacteria just replicating, right, and reproducing that way. But often people forget that large multicellular organisms actually can reproduce clonally as well sometimes, as a normal case. Okay? As opposed to sexual reproduction, where there are 2 parents, right, and you have unique new combinations of genes coming from those 2 parents. So first, an example of clonal propagation. Right? So this one is a hydra, and you might be familiar with this. It's a small aquatic creature. It's in the group called the Cnidarians, and it normally reproduces most of the time by budding. Okay, so a small individual will just grow off the side of an existing one. Okay, and that's how it reproduces. When times are bad, it can actually reproduce sexually. So if the water quality starts to go downhill, it actually will make gametes and reproduce sexually. But most of the time, it just reproduces clonally. And in fact, in some laboratory populations, they're only known to reproduce asexually by clonal methods. But then other things that you might not be aware of are also doing this. So when you see a stand of redwood trees that's like in a circle, okay, those are actually the same tree. So the roots are sending out runners, and new trees are coming up. Okay, and they're actually the same tree. They didn't reproduce sexually when they did that. And a famous example of that is this forest I think it's in Utah, aspen trees. Okay, I think it's called Pando, and it's about 47,000 trees. Okay, they're all male. They're about 80,000 years old, and they weigh something like 6.6 or 6,600 tons. They are actually just one tree. Okay, so the tree is just over 80,000 years expanded under the ground and sent up runners and sent up new treelings, but it is actually just one tree. Okay, so it's thought to be the largest organism in existence. Okay, and so it's genetically identical. All of those 47,000 trees are actually the same. All right, and that's actually one reason why they all turn color at the same time, because they're actually one organism. All right? Yeah. It depends how you want to think about it. So, I mean, people used to always think of them as separate trees, right? But once you realize that they are actually still connected to each other in a clonal, you can separate them, and they do become separated. The roots become separated, but they still are clones of one another. Okay, does that make sense? So it'd be like thinking about if you had a bunch of bacteria, you still think of them as individual bacteria, right? But they are genetically identical to one another. And so the same thing with this entire hillside of trees. Okay. Okay. So then the counter to that, of course, is the sexual life cycle, where you have fertilization and meiosis alternate here. Okay. So in the case of humans, right, your somatic cells, right, so most all the cells of your body, other than the gametes, right, they have 23 pairs of chromosomes. That's the complement in humans. For other animals, the numbers are very different. So, for example, Drosophila have 4 pairs of chromosomes instead. Okay, and they're in pairs. You received one from your father and one from your mother. Okay, you have 23 pairs. The karyotype, and you've seen pictures of this already, right, is an orderly display of the pairs. So you see them. Generally, people do this by catching cells in metaphase and then sort of smashing them open and then looking at the pattern of chromosomes. Because remember, chromosomes normally aren't condensed except when you have replication. So that's the only time you're going to see them as nice chromosomes. And the 2 chromosomes in each pair are called homologous chromosomes. Okay, or homologs. Okay, and chromosomes that are homologous are the same length and shape and carry the same gene. So they're the copy that you got from your father, the copy that you got from your mother. They have the same genes on them. Now they can have different alleles of those same genes, though. Okay. And then, again, just emphasizing for a homologous chromosome pair, one of them comes from your father, the other comes from your mother. All right? So then this is a picture of chromosomes from a human cell. All right? But again, emphasize that this cell was in metaphase. So what you're seeing here are sister chromatids together. Okay? So this one are the 2 copies. There are 2 copies. You had originally gotten one from one of your parents. You're now undergoing DNA replication, so you've made 2 copies, and those sister chromatids are still stuck together. And then this one could be, say, from your father, but you've made 2 copies. And then this is just a nice layout. They don't look like this when you smash the cell. They're all randomly arrayed. Someone's cut the little pictures out and lined them all up. So you can see that you have 23 pairs. Okay? Okay. There's one pair that's a little unique, and those are called the sex chromosomes. Okay, not all animals have sex chromosomes. We'll talk about this. Humans do, flies do. Okay, and these play a role in determining the sex of the organism. In the case of humans, we have an X and a Y, and so human females have 2 copies of the X, and human males have 1 X and 1 Y. Okay, and the other 22 pairs of chromosomes we call them autosomes. So you have 22 autosomes, and then the 1 pair of sex chromosomes. Okay. And each pair of homologous chromosomes, again, includes 1 chromosome from each parent. So the 46 chromosomes in a human somatic cell are 2 sets of 23, right? 1 from the mother, 1 from the father. We keep repeating this just to make sure that you guys get this, right? So we say that a diploid cell, we call that 2N, and it has 2 sets of chromosomes. So most of the somatic cells in your body, right, we call 2N because they're diploid, meaning that they have 1 chromosome, 1 copy of a chromosome from your mother and 1 copy from your father for each of the 22 autosomes. Okay, and then also for X and Y, actually. Okay, so that means that in humans, the diploid number is 46. So 2N equals 46. Again, so you have 23 pairs of chromosomes with 46 chromosomes. Okay. And then when DNA synthesis occurs, right, each chromosome is replicated and they sit as the sister chromatids. We went over that when we went through mitosis. Okay. So a cell, if we draw it like this, so again, we're drawing a cell that's already undergone replication. So in this case, it's prophase or prometaphase because they're not lined up yet. But you see that they're sister chromatids. And so we're making up an example here where there are 3 chromosomes. Okay. And we'll call them, we'll just arbitrarily number them for the moment so you can follow them. So let's call the longest one 1, the middle sized one 2, and the shortest one is 3. Okay. So you can imagine, say, that the blue ones are is the DNA information that came from your father, and the red one is from your mother. Now they're already replicated. So remember, in a normal somatic cell that's not in mitosis, you just have 1 blue line here. Okay, but the homologous pair are these 2. Okay, so they have the same genes on them. Okay, 1 came from your mother, 1 came from your father. Okay, so in this case, the N is 3. Okay, but 2N, the diploid number is 6. So that means that you've got 6 chromosomes. You should, like I said, go through this carefully in the book and make sure you can follow it. Okay, but the gamete, an egg or sperm, is haploid, so it only contains an N number of chromosomes. Right, so it only contains 1 copy of those. Okay, and so for a human, then the haploid number N is 23. Okay, and that set is going to be 22 autosomes and 1 sex chromosome, either an X or a Y. Okay, and because females are XX, that means that an unfertilized egg from a female is always going to have an X chromosome as its sex chromosome. But for sperm, because males are XY, okay, and then the sperm ends up with 1 of those 2 chromosomes, it either ends up with X or it ends up with Y. All right? Okay. So then thinking about the life cycle, right? So fertilization is the union of gametes when sperm and egg combine. Okay, and the fertilized egg is called a zygote. Okay, so you have the gametes, fertilization occurs, and you have a zygote. And the zygote is, again, what's going to produce all the somatic cells. So the zygote is 2N. So the zygote is returned to a diploid state because each gamete is haploid, but the zygote is formed by a fusion of those sperm and egg. And so now you go from N to 2N. Okay, but again, now one of them came from your father, and 1 is coming from the mother. So 1 set is from the sperm, 1 set is from the egg. Okay, all right. So how do you get the gametes, though? So at sexual maturity, the ovaries, and we're talking humans in this case, right? At sexual maturity, ovaries and testes begin to produce haploid gametes, okay? And it does it by a process called meiosis, which we're going to go over in some detail, okay? Rather than mitosis. So meiosis is the process by which you can actually go from a diploid cell into a haploid cell, and that's what you need to do to make sperm and egg. So it results in having only 1 set of chromosomes in each gamete instead of the 2 sets of chromosomes, okay? And fertilization and meiosis alternate in the life cycle of most organisms, right? Okay, so in humans, again, your somatic cells are haploid, but you undergo this process of meiosis, and you end up with your somatic cells are diploid. You undergo the process of meiosis, and you generate egg and sperm, which are haploid. Fertilization returns you back to a diploid state. Okay. We'll end just by telling you that there's, though, a variety of sexual life cycles, and you can get this alternation between meiosis and mitosis. So in humans, the haploid state is very transient. It's just the sperm and egg, right? But there are other animals that spend a lot more of their life in a haploid state. And so we'll just show you an example real quick. So again, we've already gone over this, that in something like humans, you don't spend the life cycle has a very short component of being haploid, just the sperm and egg, and they fuse in order to make the zygote, okay? But plants and algae can exhibit an alternation of generations where they actually alternate between diploid and haploid multicellular stages, okay? And the diploid organism is called a sporophyte, and it makes haploid spores by meiosis, okay? And each spore grows by mitosis into a haploid organism called a gametophyte, okay? I think what we'll do is we'll just pick this up next time. All right, thanks.